Hello, my name is Ben Templeton, and I'm a fourth-year undergraduate biomedical science student here at The Ohio State University. For the past 2 years, I have conducted research in the BC Adeki Lab within the Department of Physiology and Cell Biology in the College of Medicine here at Ohio State. Our lab looks at ways we can post-translationally modify, through methods like phosphorylation, different proteins within our heart to improve cardiac function. There are two major muscle filaments that function to interact with each other to contract and relax our cardiac muscles within our heart. The thick filament, one of the two muscle filaments, is called myosin. Myosin consists of a variety of heavy and light chains and protein bindings molecules that allow it to interact with actin and contract our sarcomeres, thus contracting our cardiac myocytes and overall heart. We also have another filament called the thin filament, or actin, which also functions in contraction and relaxation of our cardiac myocytes. On actin, we have a variety of different proteins that can affect the interaction between myosin and actin. One of the major muscle or proteins on actin is called tropomyosin, shown here in blue. Tropomyosin is in its sterically blocking site in this diagram, showing a relaxed cardiac myocyte because it's sterically blocking the myosin binding site on actin, thus inhibiting the interactions between these two filaments and not allowing contraction to occur. Also on actin is the troponin complex, which is another regulatory protein that affects interactions between actin and myosin. There are three main subunits to the troponin complex. One being TNC, which is what holds tropomyosin in its myosin binding blocking position, as well as TNC, which is where calcium will bind to trigger the contraction of our cardiac myocytes. And finally, TNI, shown here in green, which will function as the inhibitory subunit of the troponin complex. When calcium is present, it will bind to TNC, and the hydrophobic ends of TNI will be exposed and allow it to bind to TNC, thus altering the entire structure here on this actin molecule. Tropomyosin will be moved out of its sterically blocking position, and myosin and actin will be able to interact to trigger the contraction of our cardiac myocytes. There are a variety of different heart diseases, though, that can affect these interactions between tropomyosin and troponin and actin and myosin. One of the major ones is myocardial ischemia. Myocardial ischemia can lead to subsequent cardiac myocyte death and is a leading cause of heart failure. During myocardial ischemia, we have elevated intracellular calcium levels that can result in the activation of a protease called calpain, which can deleteriously cleave TNI at its amino terminus. However, there are ways we can modify TNI to potentially limit the harmful effects of this cleavage by calpain. And one of the major ones is phosphorylation. Previous research has found that the phosphorylation of TNI at the amino acid residues serines 23 and 24 can reduce the cleavage by the protease calpain. My project is looking at if TNI can be phosphorylated at a different amino acid site called tyrosine 26, which during ischemia could potentially also limit the harmful effects of the calpain-induced cleavage by the protease calpain. So, to summarize the objectives of my research project, we are looking to determine the role of phosphorylation at the tyrosine 26 site on TNI in the regulation of calpain-induced proteolysis, and we're looking to determine if these results hold up across three separate models of TNI by itself, the entire troponin complex, and troponin isolated from cardiac myocytes. And we hypothesized that tyrosine 26 phosphorylation will reduce calpain cleavage similarly to serines 23 and 24 across all three models. So during this experiment, we will be conducting a variety of calpain cleavage assays of both purified TNI, the entire troponin complex, as well as what we are now working on, which is myofibrils isolated from cardiac muscle tissue. And we will conduct Western blot quantification and imaging across all three models to determine if the TNI susceptibility to calpain is being reduced. So, first, we conducted models using TNI by itself, shown here in this upper square. So, we used three different varieties of protein to test our hypothesis. First being TNA wild type, then TNI Y26E, and then TNI S2324D. We also conducted 5 different time points across these models: 0, 1 minute, 3 minute, 4 minute, and 8 minutes across all 3 proteins, using 0.02 units of calpain per microgram of TNI for each sample. In figure A, we have Kumasi stains showing the change in protein concentration after being exposed to calpain across each certain time point. And then we have a quantification image shown here in figure B, where we see wild type in blue has much higher levels of cutting across almost all time points compared to Y26E and S2324D, illustrating that the cleavage by calpain is being reduced using this phosphorylation. Now in this lower quadrant, we conducted a similar test as in this part. However, we did this using the entire troponin complex, not just TNI by itself. So again, we exposed it to calpain, this time diluted 1 to 25 at 0.02 units of calpain per microgram of TNI. And the concentration change was measured across time points 0, 15, 30, 45, 60, and 90. Here we have 2 representative westerns. Here comparing wild type and S2324D, and in figure B comparing wild type to Y26E. Both here are showing reduced cleavage between the S2324D and Y26E time points compared to wild type. And this quantification graph further illustrates that point, where across multiple time points we see reduced significant cleavage in our phosphorylated protein TNI complexes, troponin complexes. Now our research is currently focused on seeing if we can replicate these models using cardiac myofibrils isolated from ventricular tissue. So our first example we would like to show is where we were testing the dilutions of different time points to determine proper conditions for our experiments. So here we had 1 to 50 dilutions, 1 to 25 dilutions, both showing promising results that we can use in further assays. As well as here, looking at if the cleavage by calpain is occurring. Here we used S2324D cardiac myocytes, and we see a downward trend of cleavage by calpain, thus illustrating that the enzyme is functioning and we are having proper conditions to allow for the protease activity to occur. So to conclude what I've stated previously, in this project, we have found that calpain proteolytic activity is reduced by the phosphorylation of tyrosine 26 on TNI at the levels of the TNI subunit and the troponin complex, and that this phosphorylation of the tyrosine 26 can play a similar role to serines 23 and 24D. And in further experiments, we will look to determine if homogenized cardiac tissue can also illustrate similar results as the TNA by itself and the overall troponin complex. Thank you for your time. I appreciate it.